こんにちは。これは外国の方もいられますね。Hello, good night, good evening. So initially in Japanese, sorry. メタフォレストセミナー、73回に、来ていただいてありがとうございます。今日は、ゲストは、私たちの最も長い、古いメンバーでもあり、日本を、ベースにして、世界のバイオアートとかバイオメディアアートを牽引してきた、ゲルク・トレメルさんに、お話をしていただきます。普段は東京にいらっしゃるんですけど、彼、オーストリアの、応用芸術工科大学なんかちょっとよく覚えてないけど、そういう機関で、今、博士号を取ろうとしておられる関係で、今、たまたま、オーストリアにいらっしゃって、そこからつないで話していただきます。で、日本語ではなく、英語で基本的に多分やるんだと思うんですけど、わからないところが、なので、多分わかんないことが結構あると思うんです。聞き取れないこととか。で、特に日本の学生さんは、わかんなくなった時に、できるだけリスポンスをしていただいて、ちょっと、ゆっくり喋ってくれでもいいし、わかんないんだけどっていうのを、意思表示を、どんな形でもいいのでしていただければ、と思います。So, this is Hideo Iwasaki, the director and co-founder of metaPhorest. And so I will have regularly an talk event called metaPhorest seminars. And this 73rd seminar will be given by Georg Tremmel and representative biomedia artists based in Japan and also in Austria. And so, could you please start?
Hello, こんばんは。私はケアです。すみません。今日のセミナーは英語で話しましょう。じゃあ、ちょっとスライドシェアリングしたいと思います。はい。どうですか？みんな見ますか？見ますですね。そうですね。僕は、okay, maybe English. Yeah? So, my name is Georg Tremmel. I'm from Austria. I have a background in media art from the University of Applied Arts here. I also studied at the RCA in London. I started the PCL, which is an artistic research framework investigating art and science, especially bioart and biomedia. And I was also working for a long time at the Laboratory of DNA Information Analysis. I think Nida-san is here somewhere in the chat. So hi, Nida-san. So it was really interesting for me as an artist to be able to be in the science operating system and work with scientists and know how the difference between scientific research and artistic research is actually happening. And of course, I'm a very long time and very proud member of the metaPhorest group at Hideo, and I think it's really an amazing opportunity, an amazing platform, an amazing group to allow artists to actually use the lab and be connected to this great group of people. I also started the BioCure, which is a biohacker space co-located with the Fab Café Shibuya. So there, it's more an open, more an unstructured space where also artists and designers can come and explore these new technologies. And of course, Hideo is also our lab supervisor. You're the scientific supervisor of the BioClub lab. And since a couple of years, I started a PhD in artistic research at the University for Applied Art in Vienna. And this is actually the reason why I'm right now in Vienna. I think maybe at the end of the talk, I will chat a little bit what I'm planning to do here or what I'm already doing here. Okay. Hideo-san, how long shall I? 40 minutes?
Up to you, but 30 or 40, and it's up to
30, 40. Yeah. Okay, so yeah, so of course I should not talk about everything, but then I realized I should just talk about the actual things. But I also realized that many things are not finished yet, so many things are in progress. And this is the challenge and problem of bioart. It's really hard to know when it's finished, because from the initial idea to the final outcome, it's really hard to judge. Maybe this is similar to performance art. How can you know when a work is finished? So I will try to interweave my works, and hopefully some things, some threads become clearer and more understanding. Maybe my English is not so understandable. Moshi boko no eigo wa sono chouzo janakute, moshi chat de iro no shizumon ma nihongo de eigo mo kaite kudasai. And if I speak too fast, which I sometimes do, please make a signal. Please speak slowly.
そうですね。
Okay.
早く、特に彼乗ってくると早くなる傾向があるんで、そういう時に
I'm excited.
早いのは、自分が聞き取れないせいじゃなくて、彼が早いのが悪いので、この場では。遠慮なく言ってください。
So just make a wave or something and say, please. お願いします。
はい。
Okay. So, but yeah, like many artists or societies, we find ourselves in a place and time where life and technology is kind of course, divided. But at the same time, technology is becoming more lifelike, and life is becoming more technology and
likely, あれ、インターネットコネクション、
closed over.
A little bit, the connection is.
Okay, so you know what? I will turn my video off.
Yeah,
Hopefully
yeah,
it's better.
Yeah.
Okay, I can hear you and I can see you. Yeah, maybe. Okay, so we also had this other quite interesting division. So when you say the 20th century was the century of the computer, maybe starting with Shannon and information theory, and the 21st century will be or already is the century of the biology. And I think this was stated in Freeman Dyson's essay about 15 years ago, but this was even before CRISPR, before gene synthesis. But just the importance of biology and the ability to change not only our environment but also ourselves. Yeah. And what I'm really interested in is how this so the computation, digitalization gave rise to digital media or media itself. And my question is, what kind of media will be possible for biology, right? So where is the media in biology? And another thing is also happening or happened in the last 5-10 years. This shift of the biological science from an analytic science, a read-only science, to a synthetic science. So before it was just called biology, right? And traditionally people observed, classified, and described, yeah? Even until the Human Genome Project. This was also just an observation and an analysis of the genome. Yeah? But since maybe 10-15 years, even the term synthetic biology became a program. And now with the ability to not only read and write DNA, not only read DNA, but also write DNA, I think there's a paradigm shift happening right now. And I think this is where I try to situate my work or our work in this shift between this from the analytic to the synthetic. And this is another exemplification. So the Human Genome Project has been renamed Human Genome Project Read, because there will be a new Human Genome Project, which will be Human Genome Project Write, with the explicit goal to create a fully synthesized DNA of a human being. But this is still ongoing. So there are many strategies and methodologies for how to create an artistic response for this. So this couple of ideas were developed together with Sarah Cook. She's a kind of media theorist during the Making Life workshop in Helsinki some time again. I will not go too deeply do it, but just to give you an idea of what things are possible. And it's also quite interesting because this strategies and methods or methodologies, they are very often they are inherent in the work, right? So it's a step back for artists to actually observe what you're doing and how you're doing and why you're doing. Yeah. So so one attempt would be to create future scenarios, science fiction, speculations, and while doing this, you can change the levels of absurdity. Yeah. And this is, of course, very much like speculative design and things like this. So where you kind of situate the story or the artwork a certain time in the future, but because of that, you are allowed certain freedoms to fabulate, to speculate, and to raise things. Yeah, but this has a long fiction, a long history. Yeah. Another interesting approach is kind of material investigations. So basically, what is the material? What is the material itself that we as artists interested in biology are actually working on? And I think the question is very much what is life itself? You know, what is the difference between the living, the semi-living, and I think many people, including Hideo and Symbiotica and many people wrote and worked on this extensively. But still the question, what is the material which we actually work on is also very interesting. Because with similar to performance art, so biology can also be seen as a sort of performance art. And I think there's Jens Hauser, okay, you don't see my screen now, but Jens Hauser, he created a very nice exhibition and show called on micro-performativity, where also Hideo's and Juan's participations are there, and where they actually try to situate bioart within a performance context. And I think this is a very, very interesting approach. Of course, with biology, you have the problem that the performative actions or agencies are very small, and how to make this visible and explorable for the visitor. And another idea is this kind of idea of tracking or exposing. So how does the situation look like? How did it change? What changed? Why did it change? You know, so of course everybody knew about the CRISPR babies, Lulu and Nana, but the surrounding things of how this got exposed, how did it change, why did it change, this is very, very interesting. And I think maybe some of my works fall into different categories of this. And of course, another thing is the parodies or paraphrasing, but this actually is very much connected to speculations and speculative fiction. Maybe I should make a nice graph for this, because I think it's quite important. Yeah, so this is basically just a summary, and I think you already can see that that things like reactions, reenactment, responses. So a lot of my works are actually can be situated in this, can be reenactments of certain historical facts or historical happenings. Yeah. So I also have to show this, because this fits also very much into the idea of the methodology. And this is from Tony and Fiona's Rabies Speculative Everything, which I'm also very happy to be part of this book with one of our older works. And Tony and Fiona, they were also one of my teachers at the RCA. And of course, they became very famous and very well known for founding this field of speculative design, right? But I think the field is very much, it's a very wide field. It doesn't have to be confined to design only. I think we as artists, we can also learn a lot from these approaches. And of course, Tony and Fiona, they did not invent this speculation. So or this kind of predicting the futures or kind of trying to map possible, plausible, probable, and preferable futures. So there are some predecessors, and actually this is one of the earliest examples I could find. So this is the same timeline or time error, if you want. But this was from a health journal from 93, right? So where again they try to predict the possible futures of health sciences, but also they had the probable, preferable, plausible, possible ones, but they also had the wild card, which Tony and Fiona did not choose to include in this. So I'm kind of curious. I will ask them next time why it's not there. but there is this wild card, this unknown futures, right? The futures we cannot necessarily predict, but maybe these are the futures which we as artists or designers can actually try to propose and see what kind of reactions this future can get. But of course, then they become maybe possible or maybe plausible. Yeah? So, yeah. Just leading into this, so earlier this year, I was part of an interdisciplinary team called the Future Panel on Synthetic Life. And this was a very interesting experience because I was invited to join this team of philosophers and very famous scientists like Sten Rasmussen and maybe Huub Swaard. He's a kind of well-known philosopher. So we were invited by the Ratenau Institute, which is a Dutch research institute which deals with the future of technologies or different kind of futures of technologies. And they invited us for a series of meetings where we actually, as a group, as different groups, we actually tried to map out how would synthetic cells, the background of this is that there's a research group called Building a Synthetic Cell, which is, I think there are many teams, I think in Japan it's called molecular robotics and things like this. So many people, many groups are building synthetic cells, synthetic life. But the questions we were asked or we tried to answer is, how would these synthetic cells, how would this synthetic life actually impact society? Yeah. So if we again speculate or kind of try to project to the a couple of years in the future, once the technology became mature, so how would this adapt our position? And I will not go over the report, but I think I just would like to point this out to you. And if you are interested, I urge you to download this and read it for yourself. But I think, yeah, what we are trying to do was mapping the social changes and dilemma in a life with a synthetic cell and kind of identify the conditions under which the construction of a synthetic cell can be considered beneficial for society. So it was not really just about synthetic cells, synthetic life in the lab, but really how would the society actually react and this. And this report is considered to be like a starting position on this question how society or societies would react to synthetic cells. Yeah. Okay, yeah, I think it's really like 30-40 pages report, so I will let you go over it if you really want to. But why I included this, because I think it was quite interesting that me as an artist, right, was invited to join this discussion. And I think this was really, I was really happy about this, because it means what we do as artists and researchers can also potentially have implications in the so-called real world, right? So what we're doing, the artistic research we are doing, can actually also be not only interesting but also fruitful for the hard sciences, right? So I think this is quite an interesting conversation that is starting to happen now. Okay, I will not go too much into detail of my works, but I think sometimes it's important to explain it a little bit in order to understand the following up works. So one work is called Common Flower Commons, which deals with genetically modified organisms and how they are received by society. So this is Santori's blue carnation. Does anybody know it? Please just raise your hands if you know. Yeah, okay. So I think everybody should know it in Japan, because Suntory is, of course, a big drink company, but it's also a big biotech company. And they developed, well, they bought a smaller company which developed this GMO, Identicomicae blue carnation. So the blue gene from another flower was inserted into the carnation, and this was put on the market, I think, in 2008. Yeah, but it's the first GMO product which is neither human food nor animal feed, but it's a purely aesthetic product, and this was a very, very interesting or very, very important meeting. Or meeting this flower was a very important event for me. And I was really shocked because there was no discussion about whether this modified flower is wanted or whether people don't want it. So there was just nothing happening around it. So I decided to create a actually work with the material of the flower itself and try to turn this very special flower into a common flower. So we did it by so this is the pathway which turns which is responsible for the blue color. And so this is, everybody's laughing now when I show this, but this was our first plant biolab, So it's the clean bench and the containers and ethanol. And basically what we did, we did tissue culturing on the flowers. And once the flower were growing, we released them into the environment. So special flower, common is futsu, right? Futsu flower, but flower commons is also the shared spaces where where actually farmers used to graze the cattle and animals. So maybe now everybody knows it from Creative Commons, right? So it's a shared land. But our idea was to actually democratize access to this plant, but also question the legality of releasing this GMO plants to the environment. Yeah? So here are some other shows. A follow-up. Okay, so this is actually at a very famous fine art museum in Cologne, and in the background you see quite famous oil paintings of. You don't see the Picasso or Miro, but they were there, and the curator was, or the director of the museum was very concerned that my plants. So it was showing biologically living artworks within a contemporary painting gallery. But as a follow-up of that, I was curious whether it's possible to actually remove the blue gene, which is responsible, the gene which is responsible for the blue color, and revert the flower back to its original state. So in the beginning, I was very much looking into RNAi, right? So everybody knows that. So RNA interference, where you insert another gene, which then cancels out the messenger RNA, basically. But then CRISPR became more popular, more accessible. So we actually tried to create a construct which removes the blue gene from the plant, and once this plant would grow, the plant would be not blue again but white again, because originally it was a white flower. So we did this, but we didn't have the chance yet to experimentally confirm that the genome sequences whether it actually really worked as we wanted it. But the bigger question, I'm curious, if we do that again, right? If we use CRISPR to remove this gene, is it actually modification? Is it considered genetic modification? Yeah, I was pretty much sure it is genetic modification, because you might also want to use some marker genes which enable selection of the plants. And in Europe, it is very much genetic modification. But there's quite an interesting thing happening in Japan right now. And I will come back to this if you really want. In Japan, since last year, there's a tomato on the market, which is called the high GABA tomato. I don't know if this is a real high GABA tomato, Sicilian Rouge high GABA. And this comes out of research from Tsukuba University and is now developed by the Sanadek seed company. And what they did, Japan approves gene-edited super tomato, but will anyone eat it? Yeah. So maybe some of you know GABA. GABA is a gamma aminobutyric acid. It's a neurotransmitter. It's responsible for yeah, for brain functions. And some chocolate has maybe high GABA content. Tomato has already quite some GABA into it, but they actually, what this company did, they removed a gene which is responsible for the control of the GABA production in the tomato. So by removing the gene, the GABA content gets higher. Yeah, and this is very important. They did not add a gene which creates more GABA, but they removed part of the DNA. And by removing this control gene, the GABA content gets higher. Yeah, so this is very, very important. Because of this removal, the tomato is not considered GMO in Japan. Nihon de idenchi kumikai de wa arimasen. Yeah, and because it's not considered GMO, it did not have to go through regulatory procedures. Yeah? So the flower, it took 5 years to make field tests and field trials to make sure it's not dangerous to human, other plants and insects. But because this gene-edited tomato is not considered GMO, it did not go through this trial. Yeah. So therefore, it was possible for me to apply and get it. So if you pay 8,000 yen, you get this box. Yeah. So we get a nice box from Kuro Yamato, and there are some nice tomato plants. Of course, then they look a bit sad now because they were in the box, but then you also become a member of a nice open chat group with Line. And then what I and many others did, we planted in our garden, right? But these are the plants in the garden, and this is the freshly planted. Okay, this is already quite old, but okay, I don't have so many pictures now. But the fact is, these plants, these modified plants are out now, right? So we can grow them. And there was actually quite some competition. So who would make the most high GABA contest? Who would make the most plants? I made seeds. Yeah. So I was taking I opened the tomatoes and I was washing them. So I have now GABA tomato seeds, and what I would like to do, very similar to the Common Flower project, is I would like to really find out what is the missing DNA sequence of this tomato, which sequences got removed, yeah, and then recreate this DNA and modify the GABA tomato again to return it to its natural original state. So the methodology is the same as with the common flower, with the whiteout, but again, questioning this state and questioning how these micro-performative actions, which the company is doing and which I'm doing, how these performative actions actually change the legal state of the plant. Because now the plant is not GMO, but if I add the missing DNA, it will be considered GMO. Yeah, because other information, other DNA information is getting inserted into it. So this is a kind of a paradoxical situation. I think this is where my work is very much situated, not by providing maybe aesthetic experiences, but by providing paradoxical situation which needs to be, or which maybe challenges, or which maybe asks the observer to resolve or to think about it themselves. Yeah. So, and there is actually, being back now in Austria, there's also a huge discussion going on about the so-called neue Gentechnik, or new genetic technology and new breeding technologies, where actually there's a big argument going on whether modifications done with ZFN or zinc finger nuclease, Talens or CRISPR would actually fall into this gene technology law or legal framework. And these differences between Europe on one side, America, Japan, and other places on the other side is also very interesting kind of meta research, like how societies approach these different challenges of how to legally define modified organisms. And of course, this also has other consequences for what's happening right now. So this is just a short chapter on the new breeding technologies, which I quoted from Wikipedia. So in a similar vein, but maybe making it a little jump back in time. So this project is called Delta Gardens. It's also an very much ongoing project, but it also deals very much with this idea of modification, being modified. So maybe some of you heard about this thing called Atoms for Bees. So this was after the Second World War. I'm not sure if it's still happening. But it was basically a program how to use atomic energy, how to use atoms for peaceful purposes. And one thing, so this happened in the 40s, 50s, 60s, right? Or maybe even before that. So one thing that happened was mutation breeding. And mutation breeding is nothing else than exposing plants, seeds to gamma radiation. And by doing that, the gamma radiation mutates the DNA of the plants. But of course, these are random mutations. So we do not know, it's not a direct mutation, but they are being mutated, right? And if you go, actually most of the fruit and vegetables in the supermarket, maybe not all of them, but very much of them have been actually produced by these random mutation events. Of course, you need to mutate and then select which one you would like to do. And it's very interesting because this is the International Atomic Energy Authority, the IAEA, which is actually located in Vienna as a kind of a neutral country. So they oversee atomic power stations, but they also oversee these mutation gardens. Yeah, and I was actually during the Kenpoku, how's it called? Kenpoku Biennale, Kenpoku exhibition. Yeah, but I think
2016.
Yeah, 2016, right? It was just one. There was no Biennale. But during that, I found out more or less by chance that not far from Hitachi, there's this place called Hitachi Omiya, and if you zoom in Google Maps, you kind of find this, yeah. And this is one of the last large-scale surviving radiation gardens in the world. So this is about like 300 meters in diameter. This is part of a model shoot I did. This is when it was constructed, right? I think it was built in 1959, and you can see very much the earth walls around it, and this is what they this is some examples of the work they did. So they were mutation breeding different colors of chrysanthemum. I'm sorry, I don't know what's the flower called in Japanese. But yeah, it's a very, very, of course, very important flower in Japan. But these colors are the result of these random mutations, right? But of course, as you can see, there's no blue because blue cannot be done through random mutation. And this is also the reason why the blue carnation was done by genetic modification rather than by mutation breeding. And here are some more expressions of this. It's very much 60s sci-fi aesthetic. And you have the gamma house, the phytotron, and yeah. And I was also lucky to get actually tour of the gamma garden, but it's very difficult to make pictures because it's such a huge space. So, and of course during the time the radiation was turned off. Yeah, but you can see the surrounding or the round features of the bark. This was me climbing up the sidewall a little bit and trying to make a bigger picture. And this is the radiation source in the center of it. So this professor is responsible for the radiation source, and yeah, it's a bit hard to see, but there's a lot of protection here, so we don't see the radiation source now. But when there's no humans, this part gets lowered, and the gamma radiation is let out, and it will radiate the plants. So okay, I don't have the close up now, but you can see there's a lot of like flowers very close to it, and they actually had radiation burn. So I could see on the leaves, so some of the leaves were actually burned because of the radiation. So I think that's always a bit the challenge of how much radiation would kill the plant, how much would just mutate it, and so on. Yeah, so this is the tower, which is quite an interesting one. This is me being very happy with a dosimeter checking the radiation. But there, of course, outside there was no radiation. But you get the radiation badge just to be sure. Yeah? But what I would like to do with this, so because this is very inaccessible, and my plan is actually now to recreate this gamma garden in Vienna. So we're very close to the IAEA. Our research institute has quite a nice garden, and I will. Okay, it's not as big as this one, but I hope to be like 20-50 meter in diameter. And what I will do, I will populate this garden with mutated plants. So which my plan is to go to Hitachi Omiya to mutate the plants there, and then take these plants and create a garden outside, well, in Vienna. So that's why it's called Delta Garden. So gamma, alpha, beta, gamma, delta. So delta is the logical conclusion, but it also means a difference, right? So how different one thing is from the other. And the idea is to also not only show mutated plants, but also unmutated plants, and then observe the difference between those. But this is very much in progress. I still need to get the okay from the university to do it. And actually, I found out during my research that not far from my university, there's the only experimental atomic power station in Vienna, which is quite interesting, and also quite interesting because one of those guys got the Nobel Prize this year for quantum teleportation. So it's quite an interesting in physics, not in biology, but in physics. So it's quite an interesting place there. But yeah, again, so reenactment, recreation, actually a bit transplantation as well. So how to recreate this historic. I very much believe this is a work of land art, right? But how to move this more to an artistic context? Yeah. So, but again, this is not finished. It's just me sharing the current progress of it. And of course, if you have any comments or questions or suggestions, I'm very, very happy to hear and to have this. But why this radiation breeding is important? I think because it is a direct precursor of genetic modification, right? So it's very important in the history of identicommunicae of modified plants, and I think it's maybe a little bit forgotten. So, and this is just a little side note of it. Also in the 70s, there was this thing called the Gammator 50, and this is actually a DIY bio device, irradiation made safe for science in the 1970s, So this is a home device for irradiating, for radiating, for mutating seeds. And basically in the center, you have a gamma source. All these round things is basically just concrete, which shields the gamma source. And you put your sample here, you rotate it, and then you get this, And this is a I think it was quite expensive, so only rich schools could observe it. But this was DIY bio in the 70s, and I always show it to. So a friend of mine, Philip Boeing, he made the Bento Lab, right? And I always show it to him, and he's also quite interested in that. And the funny thing is, these gammators, they got lost, right? So maybe not many people used it. But in the 90s or 2000s, there was a bit of a scare because the radiation sources were still active, and they were somewhere in the school basement, right? So actually, the American government really tried to collect them, and I'm kind of working with Philip, so we want to maybe also recreate this Gammator maybe as an add-on to the Bento Lab. So it's a bit of a funny add-on, but it would be nice to have this in like a small version next to the Bento Lab. Yeah. Okay. Yeah, again, so the current status is I'm preparing the garden site, which is of course mostly bureaucratic work. So whether I'm actually allowed to have such a huge part of the garden for a couple of years. So I would like to collect and breed mediated plants. I will radiate the seeds in Hidachi Omiya. And of course, the most important thing is actually planting the garden in next spring. And I think also the most difficult or the most challenging part will actually be how to observe it, So I'm really thinking much right now how to observe the growth, how to measure. Maybe we can have small or maybe natural gamma radiation sources in the garden. So yeah, this is also very much in progress. Okay, so maybe I will. Okay, I will skip over this. If there's interest later, I will go deeper into this, but I just mention it briefly so we know, so you heard it at least, right? So this project is called the Blacklist printer, which is again situated between the analytic and the synthetic part. So the idea is, or the background is that there is a DNA blacklist which considers, this was actually first done before Corona, right? So please take this in mind. So there's a list of forbidden DNA sequences which produce harmful, toxic, or dangerous materials. Yeah, and there is this list of these organisms of dangerous DNA, but this is considered secret, right? Because why? Because if you order DNA and you want to have whatever, the best bacteria or some other dangerous DNA piece, you normally would not get it. Maybe if you work in a research lab which does research on that, then there's an exception. But usually the DNA sequence company would not synthesize this DNA for you. Yeah, it maybe would actually call the authorities and say that maybe you might be a terrorist and do something dangerous. Yeah. But the fact that this list exists and that is interesting. So right now, DNA synthesizers are centralized, right? So I don't think at Waseda there's a DNA synthesizer. Or maybe one with very short DNA sequences. But now some startups actually creating cheaper oligosynthesizers.
Definitely we have, because we have a department of chemistry. So they are very high level organic chemistry, and they have.
But you would not order DNA from there, right? You would order it from a company.
No.
Yeah.
That's true.
So, but some companies are actually creating like desktop synthesizers, which might cost maybe $5,000, where you can actually synthesize the DNA on your desktop, maybe similar like to a kind of high-end DNA printer. But basically what that means that one more thing. So the big DNA synthesize companies, they have this blacklist. They check the incoming DNA. They will not synthesize it if it's a forbidden DNA, right? But if everybody has their personal synthesizer, how can we make sure that people don't use it for dangerous purposes? Yeah? And this is the question which I'm actually trying to explore with this. So I made this, of course, is not functional, but it's a mock-up of it. And it's a mock-up DNA synthesizer which at some point should synthesize the DNA, and this DNA would then be dropped on top of the paper and stored as it. But of course, the DIY synthesizer also creates a lot of errors, right? And the goal is to improve the synthesizer during exhibition, during the next couple of years. But the question is, do we actually want to improve it? Do we want to give this power of DNA synthesizer in the hand of the public, right? Are we mature enough to do this? So this is a kind of a comment on this border. So this was shown in the Aomori Center for Contemporary Art. Okay, so maybe I'm starting to wrap up now, but this is another thing I would also like to show, because it's one of those projects which is not finished, and it's not finished because of like moral or ethical challenges which arose during the project. So I think most of the people here are familiar with Henriette Lacks and the story of the HeLa cell line. If you're not familiar, very quickly, so Henriette Lacks was a Black American woman who suffered from cervical cancer. Unbeknownst to her, a sample of her cells were taken by a doctor and kept in the lab. So a couple of years later, Henriette died actually, but the cells, because they were cancer cells, actually kept on living, or they did not die. Yeah, because the cancer cells had certain mutations which did not allow the cells to die. Usually with cell cultures, cells divide like 20-25 times, then cells die. But these cancer cells did not die. And since 1951, these cells, this cell line, have been a stable, very important, a very important contributor to medical research all over the world. Yeah, because just by the fact that it can be kept alive. Yeah. There are very interesting books about it. There were also very interesting artworks by Oren, Symbiotic, and other people. And this is a very interesting and also very problematic story, like how the family of Henriette Lacks didn't get recompensated. Who is the owner of the cells? Yeah, but with my work, I would actually very much highlight this question. Another step back is, this is a macaque monkey in Borneo, and he took this picture, right? The monkey took this picture because the photographer went to the jungle, he put the camera, monkey came, took the picture. The question is, who has the copyright of this picture? The photographer says it's his copyright, yeah? But he didn't take the picture. The animal rights activist said, the monkey has the copyright of the picture. And it actually went to court, and it turned out that the monkey cannot have the copyright because the monkey is not a human being, right? So only humans can have copyright of images. And this works very simply. So whoever takes the shutter, if it's an iPhone or a camera, the person taking the shutter has the copyright. So what if we can find a way that the HeLa cells, or HeLa herself, can operate the shutter? What would happen if we would make an experiment where the cells would grow, their growth would trigger the shutter, and the camera would make a picture? Who would have the copyright of this picture? So this is the Argus C3. It's a very simple camera from the very simple and very common camera from the 1950s. So actually, I got hold of one, and I created this this mock-up of the thing where in the glass container the cells would grow. They would trigger a wire, and this wire would take a picture. So sometimes I come to Waseda, and we have the HeLa cells. We can revive them. We can grow them. We are now working very much how to grow them in 3 dimensions to actually have a better control of this. And I also have been working, and there are other challenges, like how to grow the cells in an incubator in the gallery, how to maintain it, and so on and so on. But these are kind of technological challenges. But what I did not foresee is that there are also a lot of ethical challenges, and these challenges are whether I'm as an artist are allowed to use these HeLa cells for an artwork. Yeah. The question is, are they allowed to use these cells for science? Or who is allowed, or instead of using, is there a way to collaborate with these cells? Yeah. And how can this collaboration actually look like? Yeah. Or do we have to ask the family for permission to use it in an artwork? but there's still very much unresolved questions about how to use human tissues or other humans' tissues without explicit consent, right? So I think this is the question. But of course, my intention with this artwork is actually to question whether or to give the HeLa cells the personhood, right? So I would argue that HeLa herself is still alive, and HeLa can have copyright. Yeah. So by doing this, by assigning the copyright to Henriette Lacks, it would mean that she as a person is still alive, and this is my intention with this work. But yeah, it is still ongoing. Of course, I could do it with my own cells. I could immortalize my cells. I could grow my cells, but then it would be a different. It would have a different meaning. Of course, maybe after after I'm dead, maybe it would be a different story that I could still use my things. And maybe again, people like Symbiotica or Guy Benary and people like that. So they also work very much in the direction of the ethical and the personhood of cells. Yeah. Okay. Okay. So this is another kind of it's also still ongoing. It's a kind of a thought experiment, which is again a recreation of the so on-off machine. So it's called on-off maybe. And again, I mentioned before information theory, and as you guys know, Georg Tremmel, he formulated the mathematical theory of communication and of information. Sorry, and he was of course a very, very strong mathematician, but he was also I think people would call him like a maker or hacker now, because he created a lot of toys, a lot of machines which did not really have any purpose. I think this is actually maybe I would even call him a media artist, right? Because he would, for example, make a tandem unicycle, like a Ichi-Rincha, right? So a unicycle, but for two people. Yeah, and the right one, this is like this jumping stick. It's a jumping stick, but he would put a petrol engine in it, right? And this was done in the 50s, 60s, 70s. So he's a very, very interesting kind of inventor. And I think actually a friend of mine made his PhD thesis on that. So if you want to know more, I have a very interesting book to recommend. And the science fiction author Arthur C. Clarke, actually, he visited him once and he found this box, yeah, and it's a very simple box, just with one button, and the button is off. But when you put the button on, a little hand comes out and turns the button off, or the switch off, right? So that's why it's called the on-off machine. And this was again done in the 50s, and I think this is a very, very allegorical work. It also represents. So Shannon was also very much working on missile defense and missile launching, and this was very much the same game, right? So the Russian-American missiles, so one would be on, and then both would be off, right? So this is a very, very interesting device, and of course, it also stands exemplary for the information age itself by having two states, by having the 0 and the 1, the on and off, right? And there is actually like a cottage industry. So many people made many different versions of on-off machines, and usually they are nice boxes, and then you have some hand-like devices which actually turn it off, you know. But I was thinking that if information theory is just between 0 and 1, then biology is the space between 0 and 1, right? So it's this between death and life, between living, non-living, this undefined space in between. And that's why I kind of die maybe, yeah, so the maybe space. And my goal is to make a little, again, a micro performance, yeah, of cells, which would actually grow. An incubator with one switch, when you turn it off, it would grow cells. I guess growing a hand is a bit difficult because the bones and stuff, but you could grow cells or living matter in the shape of a hand. And this hand, again, would switch the machine off. And by switching it off, it would also turn off the incubator, right? And its life support. And the cells inside the incubator would actually die. So basically a biological interpretation of Shannon's ultimate machine. But again, this is still speculation, so I still need to learn more about 3D tissue culture, but I hope I can be at a level where I can do it very soon. Yeah, how are we about time, Hideo? Shall we stop or shall we?
Yeah, you already explained a lot of things,
Okay.
but if you have a very ongoing project with Vienna,
Yeah, okay.
could you briefly add?
Yes, okay.
Or this one, or
Yeah, so very briefly, so one of our previous works was Ghost in the Cell, where we created synthetic DNA, which was for Hatsune Miku, was then transferred into iPS cells, which were then induced to become heart cells. And these heart cells with the Hatsune Miku synthetic DNA was actually living and growing within a gallery in Kanazawa. Right? Earlier this year, I was invited to go to Guangzhou, to the Asian Culture Center, which is an amazing, fantastic art space, and it's very new, very big. And but of course, they didn't have much support for bioart. But I had another. I'm working. What we're doing for there now is actually to. So if you have this heartbeat video of the cells, we are creating AI-mediated remixes of this heartbeat for the exhibition. So we're combining the visual data of these images. We're combining the so actually for the exhibition in Kanazawa, we already did some deep dreaming. So we will combine this. We use the heartbeats as source data and combining it with the actual DNA, yeah. So we're gonna remix the networks, and we will create synthetic heartbeats, which are a synthesis between the DNA and the images, and they will be shown next to the original heartbeats in the gallery. So it's more like it's not a wet bio work, but we use the DNA which has been created before. And which will be shown there. So it's a remix of the work. So, but it's quite interesting to go very deep into networks and try to create to put the DNA data and the image data on it. You know, so it's it's very much in progress. So I can share. And the exhibition is going on until next year, so it will also change during the exhibition. Okay, maybe I'm gonna skip that. So what I'm doing in Vienna, so I was very happy to be awarded a short-term or like a research project called Biological Interfaces, Ludic Cultures and Non-Human Agencies, Games of, Within and Beyond Life. And this is a collaboration with Margarete Jahrmann. She's a professor for experimental game cultures and also kind of a very well-known media artist. She studied with Roy Ascott, and she's a very important figure in net art. And I'm very happy to collaborate. And she kind of termed this term, this ludic. So ludic meaning playful or kind of experimental, and we're trying to combine these ludic approaches with biological approaches and question how non-human agencies, may they be bacteria or human cells, can actually be put into a state of play or exploration. So we got a budget of a couple of €1,000, and but again, this is an art university, and it's sometimes difficult to do things straightforward. So we will hold a series of bio-wet workshops at the university. But for these workshops, we need machines and devices. And hopefully at the end of the workshops, we will actually have a functional lab at the university, which then can be used for other artists and other works. So this is why I'm here now. So I got many meetings next week, and I will share the plans once we are there. So if you're in Vienna, you're very much welcome to join the workshops. If you're in Tokyo, maybe next year, the Biohack Academy will also be very much informed by what we're doing here. And yeah, of course. So that's it. So thank you very much to metaPhorest, to the Angewandte, and of course to the whole BioClub community. すみません、結構多いだった。でももしは特別の質問あれば、I still have many more slides to show, but ごめんなさい。僕の問題はちょっと I cannot condense. Yeah.
So do I. Thank you very much. A great talk. And so probably you showed already about 10 projects, and it's full of very informative interest. And I think that George always managed to capture the ambiguous, ambivalent aspects and the unsolved conditions related to the social reception of biotechnology. In his work, and considering, especially, it's interesting point, considering its very interesting historical context, and so it's sorry in Japanese, 多分早口で結構矢継ぎ早にプロジェクトを紹介したので、ちょっと初めて聞いた方はなかなかキャッチアップできなかったところもあるんじゃないかなと思うんですけれども、何かコメントとか質問とかあったら、チャットでも何でもいいので書いてください。Okay, so if you have a comment or the question, please. And there is a question from Ryoji.
Endo-san, you want to talk? どうぞ。日本語も大丈夫。英語頑張ってください。
そうですね。どうしよう。なんか、チャットには英語で。
There are guests from abroad, so it might be better to
Japanese.
In English.
In English. Okay. Okay. Thank you very much for your great presentation. I'm interested in the radiation project. You went to go to the Ibaraki. And then I have two questions. Actually, in the radiation project, you mentioned in the middle of lecture, are there any medical ethics issues involved, like HeLa cells? This is the first question. And then also, this is a characteristic, the radiation project. And in that, I think that really scientific and guarantative figures will be an important perspective in terms of whether or not actually you are this is the exposed to radiation or not. So do you have any hypothesis for your project from a bio-art perspective?
Yeah, of course, the radiation project. So just to remind you, so when I was in the garden, the radiation was turned off, right? So there was no radiation. So when they turn radiation on, they block it very much, right? So there's a lot of safety. There's a lot of safety, right? And of course, I cannot take a very powerful radiation source and put it in my garden, right? This is not possible.
Right? I see.
Because it's very dangerous, right? So if you're there, if you got this doses of radiation, you would very likely develop skin cancer or other cancers, right? So like if you go to the Röntgen, X-ray, Röntgen, right?
Yes, Röntgen, yeah.
So you got this protection to not. So it's very, very dangerous. And of course, I do not want to work with dangerous material, but I'm interested in the effects of radiation. So that's why you can go to Ibaraki, you can ask them, please radiate these seeds, right? And they put it in a radiation chamber, and they do this. And actually, but there are some stones actually have natural radiation, or like background radiation. You can actually detect it. And also maybe the fire, like the fire sensors in the house, they also use a very small radiation source, actually. So I plan to put some radiation sources in the project, but they will not be dangerous. Yeah? So they will be natural, harmless radiation sources. Yeah, and actually there's a story that also banana peels, yeah, if you collect a lot of banana peels, they also give us up low radiation. Yeah. And this is why back in the future, they put banana peels in the DeLorean to power it. Yeah. So, yeah. But of course,
I think, sorry. Yeah.
No, it is an experiment, but with all these experiments, we need to be very careful. I have no interest in harming myself or others. So.
Oh, I see. Thank you very much for your answers. Actually, I just think of about the DNA problem something. Actually, the DNA was founded in the past. Yeah, maybe there are a lot of people who think it's really dangerous things, but maybe it's near to the like banana stone, I think. So we have to tell the fact, and it's very difficult problem. So thank you very much.
Yeah, of course, the story with Rosalind Franklin, and actually, Hideo, I didn't know that your parents were also radiation scientists. Where did I read this? I read it somewhere recently. So the famous picture of the DNA that Rosalind Franklin took was also X-ray crystallography, right? So where DNA and other substances are crystallized, and then through the refraction patterns of X-ray, the structure was elucidated. So this is a very, very also historical part of it, right? And Rosalind Franklin, she died because of being exposed to the X-ray.
I see. It's more historical, than DNA. Thank you very much.
Okay.
Okay. Thank you for the question about the metaPhorest. I'm sorry that. So George, is this still working?
Yeah. So
from net to org. Okay. Sorry.
Yeah. So we're in the process of redoing the metaPhorest org website. So the information is there. Maybe some links are broken, but hopefully very soon we will do a relaunch or
anyway, it's a very old one, and so and we
need new one.
So we are missing 7 years. Okay, sorry. Very new.
Yeah.
But anyway, so it's on a platform set in our institution in Waseda University in Tokyo, and in 2007, and so it's about a 15 years anniversary, and we have accepted the artists and the designers, and again more than 25 artists, including our George, and so we are running a so-called long-term partnership with artists, not a very short artists in the residence. For example, George have been a member for more than 10 years, and so like this, and we how can I say, recommends the long-term relationship between the scientists and the artists, and basically it's about microbiology lab, and some artists use actually biological benches, but not so, we accept any type of artist who are interested in biology or bio related issues including philosophies and some humanity studies, and so on. So, they do not need to use the biological labs actually. And so in this sense, it's not very strongly organized artist in the resident program like Symbiotica, but like a more lazy community. like that.
So what is amazing is that Hideo, it's a research lab, right? It's a microbiological research lab working on circadian rhythm, on cyanobacteria. But Hideo, Professor Iwasaki, he very kindly opened the lab to artists, designers. So we can actually use, of course, we have to learn how to use the lab. So nobody, it's not a residency where some people do things for you, right? So the artists need to learn how, of course, you can ask for advice, but you need to learn how to do this. Yeah, you need to learn how to use this. But still, this is a very, very amazing and unique thing that it's possible to actually be able to use the facilities and devices, machines, and also the great community within this lab. Yeah.
Thank you.
And also many of the students from Hideo also they became artists. So although this is not an art university, so this is also quite interesting.
Yeah, thank you. Any other question or comments? 日本語でもいいですけど。
Okay. I have a question. George, thank you for your presentation. It's very exciting. And your DNA Blacklist printer is about technology monopoly. というか、すみません、ちょっと日本語でちょっと後半からいきます。ごめんなさい。多分そのテクノロジーっていうものを、ある種、そういう企業であったりとか、国っていうものが、ブラックリストに載っているような DNA の、なんて言うんでしょう。保持したりとか、交換したりっていうのは、そういう、国とか企業が管理しているっていうことに対して、ある種、批判的な行いとして、DNA ブラックリストのプリンターの作品があると思うんですけども、こういうその批判的な作品っていうのを見るときに思うのが、例えばそういう制度とか、バイオテクノロジーの批判、バイオテクノロジーの独占みたいなものに対する、態度っていうのは結構その国によって違うのかなっていう印象がありまして、例えばアメリカとかっていうのは結構そういう911とか炭疽菌のテロとかがあったので、テクノロジーに対するリスクの考えが強いので、結構そういうバイオテクノロジーを誰が使うのかっていうことは結構センシティブな話題になってるっていう印象があります。でも日本の場合はバイオテクノロジーが起こして何らかのそういうの、何か問題っていうのはあまりないので、規制とかに対してもあんまり強いイメージを持ってないのかなっていう印象があるんですが、作品を展示したときになんか国によって反応の差が結構顕著に分かれるとかっていうようなことはこれまでありましたかっていうのを英語で言いたかったんですが、すいません、そんな感じです。
Okay, so if I correct you, of course, there's differences in countries, how these technologies are approached, and you rightly mentioned the 9/11 and post 9/11 responses of American government to this, and of course, there's also quite a long history of that. And when the first biohacker spaces started, maybe 10, 15 years ago, there's a mailing list, right? And then there was an invitation from the FBI to come to a meeting, right? And I saw this and I thought it was a joke, yeah? But many of my friends went there and the FBI wanted to meet what kind of people the biohacker people are, right? So are they dangerous? Are they friendly? Are they bad actors? And this is actually one of the reasons why most biohacker spaces try to be very open, right? So there's nothing to hide, right? And of course, also one of the people there was David Kong, and David Kong, he's now running the MIT Bio-Community Initiative, and he's also running the Bio Summit, which I'm also a co-organizer. So we will make a call for participation very soon. So if you want to know more about that, please join the Bio Summit end of January. I think you're going to make it. But of course, it's different from country to country. And in Japan, there's a little bit of a hidden history because the Japanese military and the Japanese so I was working at Ikeken, Ikekubiken Yujutsu. And this was before called Institute for Medical, now it's called Institute for Medical Science, but before it was called Institute for Infectious Diseases. And this institute also played some role during the Second World War to develop biological weapons. Right? I have another work for this, which I didn't talk about today, called Resist Diffuse, but this is also very much, and after the war, those scientists were got by Russia and by America, which were developed into biological weapons programs in both sides, right? And the Blacklist is it's not a monopoly, it's a biosecurity measurement to ensure that bad actors cannot create biological weapons. But of course, now this is getting the borders blurry, you know, and this is my comment to that, right? So.
OK, thank you. わかりました。特にこれもごめんなさい、日本語で申し訳ないんですが、日本のその二次大戦の時の生物兵器の話とか、確かにゲオルグさんのおっしゃる通りだなと思いました。ありがとうございました。
Hi, thank you very much for your question.
Thank you very much. And so actually, and it's as conditions, as a national condition is quite also different in the biology itself. And I am, of course, a microbiologist, and I usually attend to the microbiology meetings. And usually in Japan, we see many doctors or medical doctors and the standard researchers. But when I attended the American meeting of microbiology or bacteriology, and many military people, military researchers are attending, and with their costume, and which are a little bit scary for me, because in Japan it's hidden in a very special space, and we do not all usually see the military costume in Japan. So yeah, but of course, so they report many military case reports in the society, and so it's very rare in Japan. But definitely, some self-defense medical institutes it must be doing similar researches, which is hidden in the standard microbiology society in Japan. So and I feel very the similar the difference, like.
Yeah, it's very interesting going to meetings to MIT, to the Bio Summit, right? So there was is always again one person from the FBI, and he's very friendly, he's very very kind, you know, and he's very engaging, community engaging, you know, but still. And of course, we shouldn't forget that a lot of funding comes from military side,
right?
Also
exactly.
also on this one, you know. So it's a very, it's a very gray area, you know. So many people are very much opposed to this, and yeah. But hopefully, there's no military funding yet in the art. But I'm not so sure.
That's a good question.
Yeah.
そうですね。まあ、訳さなくていいですかね。大丈夫ですかね。はい。So Olga, could you please tell the question and
okay, so I read it. So Olga, I'm not sure if you can speak, but okay. So the tomato project, did you exhibit it somewhere? Okay, no, the tomato project is still ongoing now. So I still got it growing in Japan. I got the seeds. And I think the next step is really to isolate the DNA and make PCR experiments to actually see the different results. But I haven't exhibited yet. But we shown the common flower commons at various places around the world. But usually I show them as living tissue cultured plants, right? So usually I don't want to show documentation or show them as archived ones, but sometimes it's inevitable because, for example, in Korea, like again 5 years ago, I was invited to show the common flower work, but only later, or kind of too close to the exhibition, we found out that the blue GMO flowers are not legal in Korea. Yeah, So they're legal in Japan, Europe, US, but not in Korea, maybe because the market is too small. So we could not import these GMO flowers to Korea. So we had to encase it in acryl and show it like this. But yeah, so I imagine showing the tomato project might have similar challenges. So, but yeah, I didn't think about an exhibition yet, but I would very much like to actually show the PCR results. So I think Nida-san is also here. Yeah, hi Nida-san. So we're working at BioClub at like a Mesonics project doing genomic analysis of meso, and I hope also to do the PCR experiment for the flowers for the tomato very soon. Yeah.
Also, and I have a question from the for the resist refuse from Olga.
Okay, yeah. So
that definitely should be related with the link to the.
Yeah. So resist diffuse was shown at a small gallery in Tokyo a couple of years ago, but also at Gedai at the University of Fine Art as part of the, how is it called? The recreate. I'm sorry, I forgot the.
No?
How was the exhibition at Gedai called last year? Where it was about the manuals and the recreation of the work. So not the work, not the works itself were shown, but just manuals and instructions of how to recreate the work. And a part of it was how to recreate biological artworks. Yeah. So this also falls into the question of how biological artworks can be archived, how they can be collected, how they can be re-performed, reenacted, which is again has many different challenges.
Anyway, and I think that the hard question was the shooting.
Okay,
when you are talking about the military related ones. So
okay,
yeah, definitely it's very has strong link to the military historical context. Yeah. sorry, in Japanese. ちょっとレフューズ、これすごく面白い、レジストレフューズって、今のリンク見てもらえばわかるんで、これは結構僕はジョージの代表作じゃないかと思っているものなんですけど、日本軍が昔、使用した、ある、なんていうのかな、感染兵器とされるものに関して、まあ、それをちょっと歴史的に読み解いて、ちょっと、また違うコンテクストをつけて、出した、ものすごくその歴史性とバイオテクノロジーをめぐる、なんていうのかな、まあ、闇的な部分と、それをどういうふうに、まあ、そのコンテクストを変化させ、アーティスティックに変化させるかっていうことを、すごく、面白い形で提示している作品なので、まあ、ぜひ、見てもらえるといいかなと思います。sorry for interruption. Yeah, but I think it's very good work. So, yeah. I really love it. Okay. Any other question? 何か、なんか、他にできるだけ若い。じゃあ、新田さん。
まあ、コメントなんですけど、まあ、岩崎先生。
新田さん、英語でいけるんじゃないの？
いや、まあ、
お願いしますよ。ちょっと外国人が多いんで、できれば、まずは英語で言っていただいて、その後日本語とかの方がいいかなと、親切かなと。
まあ、So I want to say just a thank you to Iwasaki sensei. So because I met Tremmel-san more than 10 years ago in the seminar by Iwasaki sensei. And so after that, so we work in Todai Ikkaken. And now so we are working Biolab about miso and genomics project.
Yeah.
So, Iwasaki-san so arrange our relationship. So,
yeah. Yeah, no,
This is very by chance. So
yeah, but of course, maybe you guys, so Hideo and Nida-san, maybe you have the most effect in my life, you know. So because Nida-san came to the metaPhorest seminar, and then you invited me to join Dodai, which was a very, very great experience. So I learned so much there. And I'm very happy that you also you're still doing your cancer research at Dodai. But you're also very much involved now to come to BioClub and also get out. So I'm very happy to have this.
So I'm a scientist in cancer research. So when I was very busy for doing research or writing paper. So Tremmel-san thinks interesting and enjoy life. I or learn a lot from Tremmel-san. So I'm so very thankful to Tremmel-san, Iwasaki sensei. Thank you.
But I'm very busy now writing paper and doing stuff, so I'm not enjoying life.
Well, I also think that Nida-san that you were running the very great workshop at my BioClub. And BioClub is, as George said, it's a DIY bio community in Tokyo. And I'm planned to join it, and I'm terribly busy, and I'm really sorry that I cannot join a lot. But it's really good project, and it's linked already. But no, this is the project to make miso and the analyze the microbiomes and in our DIY style, and it's very democratic, and I really appreciate his contribution to the DIY Bio Society in Japan a lot. Thank you very much.
Yeah, thank you very much for your support. Yeah.
Yeah, so just a bit of context. So because actually the metaPhorest lab, it's difficult to get in, right? So because it's open, but still it's a research lab. So we made the BioClub to be more accessible, more and easy, where more experiments, where we don't disturb the PhD researchers too much, right? So this was the motivation for creating the BioLab, and it's very much the same community, and I'm very happy many people are actually here on the call as well. So you got Hasegawa-san and
ということで、なので、ミソミックスのリンク、知らない、sorry in Japanese again, and I'm because the young participants do not know much about the DIY bio or some the bio club events, so I will briefly introduce them in Japanese. ということで、DIY バイオって、いずれ、なんか生物学史でも、生命学でも多分取り上げていく項目だと思うんですけど、生物学の民主化といって、今まで企業とか大学とかに独占されてきた生物学の研究というのをもっと、多くの人に、開いていって、自前で、なんかもちろん安全性とかも学びつつ、もっと自分たちのディマンドに、沿ったような形でもっと自由に、アウトプットも論文とか製品とかだけじゃなくて、アートとかだけでもなくて、もっと自由に、扱えるように、しようっていう、ある種の社会運動だと思うんだけど、それを、実践してきているんですね、私たちが。で、ゲオルクはその旗振り役の一人で、その渋谷にバイオクラブという、プラットフォームを実際に作って、もうここに参加者も何人かいますけども、いろんなタイプの背景を持った人が集って、生物学の実験とか自分たちのアイデアでこういう企画が面白いんじゃないかっていうことを考えていきながら実践するって、考えるだけじゃなくて実践するっていうところがすごく重要なところだと思うんですけど、で、その時には、科学者と、専門家じゃない方がいろいろと集って一緒にワークショップをやるっていうことがすごい大事なことで、それでニーダさんがちょうど、味噌を科学するワークショップっていうのを開いてくださって、味噌ミックスっていい名前ですけど、 味噌は確かにすごく面白いターゲットですよね。だから日本の伝統的な食品でもあって、私たちにすごく身近だけど、同時に発酵のの、なんていうのかな、代表的なものなんで、それをモカバイオの目も通して、自分たちの生活の含めて捉え直してみるっていうのはすごくいいターゲットだなと思っていて、素晴らしいプロジェクトだと思っているんで、もし3年生の方とか4年生の方とかでも、できたらぜひ一回なんか覗いてみてほしいなと思っていて、で、今、木曜日とかにやってるんですかね。火曜日？
はい、木曜日に、はい、やってて、11月の5日にちょっと実験を、16S シーケンシングをこれからやるんで、DNA 抽出してチェックをするっていう、PCR でチェックをするっていうんで、
そうですね。
よろしく
なので、結構、ある意味ではラボでもやってないような、こともやっているので、すごく普通に勉強にもなると思いますし、そこで、出会う人々がとてもユニークな人たちが多いので、面白いと思いますので、ぜひ、機会があったら参加してほしいなと思ってます。
Yeah, please join our Discord. I put this link in the group, in the chat here. そう、そっちでもうミソミックスの。そう、普通は毎週火曜日の夜は普通のバイオクラブミーティングと毎週は木曜日は、大体オンラインもそのミソミックスの特別のミーティング。詳しくはその Discord でチェックしてください。
ありがとうございます。Thank you very much.
ありがとうございます。
ということで、any other comment は、なんか、他にもなんかいろいろと多種多彩なメンバーがいるのです。森岡先生とか。
comment, but I just see Henry is also here. Hey, Henry. Hi, Henry.
Henry.
Yeah, so Henry is also a very great artist from Thailand. Where are you now in the world?
I'm back.
Henry came to BioCamp workshop that we did like 3-4 years ago, and Henry made a great workshop, a residency today, this year in Naresuan University in Phitsanulok, where he expanded his own residency. So instead of doing a residency alone, he invited 30 or 50 artists to come and join you at Naresuan at Baksa at a fantastic synthetic biology lab. So he started with Drew Andy. So the professor there started with Drew Andy. It was really a great experience. And you have plans for Singapore, right?
Yes, right now we are planning to second phase in Singapore. Yes, it will be in March, 15 to 19 March.
Yes, hopefully we can join you there. Very much looking forward to that.
So am I.
Yeah. Yeah. So sorry for putting you on the spot.
Thank you.
Yeah.
Any other question or comments? 桜木さんなんか出してたけど、なんか、あったりしますか？
いや、私はただ単にヘンリーに手を振っただけです。すいません。
You just wanted to hi to Henry. Okay. はい。
もう一つだけ質問してよろしいですか？ Can I have more questions?
もちろんどうぞ。はい。
I have a question about HeLa, your work. If Henriette cells still her body, can we say that Henriette is focused to take picture? 要するにその、
わかった。Yeah.
Okay, sorry. Okay.
No, of course, and this is the question of agency, right? So she has the cell, she has the living matter, but the question is, do these cells have agency and free will, if you want, or direction, right? And this is, of course, not answered, right? And of course, it's very difficult to answer. But yeah, but just that's why I have this little shortcut. Just the expansion, just the growth of the cell is actually triggering the shutter, right? So, but I think this is a very, very the question of agency is a very important question, And this is what I would very much like to focus on my research here in Vienna, like the question, the agency of humans and non-humans. But yeah, thank you very much. But yeah, I cannot answer, right?
Okay, okay, thank you. And I interesting your Vienna next project. そのプロジェクト話もまた聞けたら嬉しいと思ってます。Thank you.
Yeah, it's still very early, so I will show once we have more information. I'm happy to share within our groups and mailing list.
Thank you very much. And 他どうでしょうか。大丈夫ですかね。森岡先生とか、どうですかっていうか、せっかくなんで。
Thank you very much for your very interesting lecture. And my my question is very, very vague, but have some connection with the Tsukamoto-san's question. What I had in mind when I listened to your topic on HeLa, cell, and camera. What came to my mind is autonomous weapons. Yeah. So yeah. So currently, various autonomous weapons have been actually using in some battlefields, and so if such autonomous weapons kill somebody, so then the question arises: who is to blame, and who actually killed those soldiers or civilians? And so it may be possible to someday place some human cells or human brain cells inside the CPU of autonomous weapons. Then those implanted brain cells do some fantastic calculations and select the correct target or incorrect target and kill those soldiers or civilians. Yeah. Then probably the question will arise: who should who should be the
responsible?
Be responsibility or ethical or legal responsibilities. Yeah. So so this is my question, but still this is very vague one.
Yeah, no.
So it's happy if I could hear your comments.
Yeah. No, thank you very much for this great question, and it's a very, very important one, right? So it's not only about who has the, in my case it's the copyright, but as you said, who has the responsibility for the actions, right? So if you have agency, you also need responsibility. And there are some artists who work with nerve cells, right? But still, if you have nerve cells, you don't have sense, right? So you don't have input output, so you don't have consciousness, right? So even if nerve cells would be there, they would still also not have, or would they have agency? Or how far can we get for consciousness, right? And maybe this also feeds into the artificial cells, artificial life. But just I put one link in the chat. It's called Project Pigeon. So the famous psychologist B.F. Skinner. So he trained pigeons also during the Second World War to act as a guiding bomb. Yeah? So the pigeons were in a bomb, and they would pick up where the bomb would fly. Right. So it's a very old biological guidance system. So maybe it would, in this case, it would be the pigeon's fault where it explodes. Yeah. But yeah, no, I'm sorry, I don't have answers. I just have more questions. But I think these are very, very important questions, and we should try to explore it together, right? Yeah. Thank you very much.
Yeah, thank you very much.
ありがとうございます。森岡先生、なんか簡単に日本語でもちょっとサマリーを言っていただけると、ちょっとついていけない人もいるかなと思うんで。
今の質問、今の質問ですか？
質疑みたいなやつを。
今の質問。
私が訳すとなんか、
いえいえ、皆さんもお分かりになったと思いますが、戦場で今、自動兵器、ロボット兵器が使われていますが、もうすでにロボット兵器が使われて、それが人を選んで殺したときに、その誰が責任を取るのか、誰に道徳的あるいは法的な責任があるのかという大きな難しい問題はすでに生じています。で、将来はそのロボット兵器の CPU の中に人間の脳の細胞とかを入れて、もっと複雑な計算を瞬時にさせるということもあるでしょうと。で、そうなったときにはさらに問題が複雑になりますが、そういうような、問題はどうお考えですかというような質問。で、それで今何でしたっけ、この生物兵器というのは昔からあって、動物を使って、そのなんかロケット、なんですかね、銃かロケットをなんかガイドするというような、そういうものも昔からあって、そういう時から頃から生じている問題ですと。非常に難しいですねというような話じゃなかったでしょうか。
ありがとうございます。すごい、特にまとめてありがとうございます。それで他に何かありますか？じゃあ桜木さんどうぞ。
はい、桜木です。Thank you for your great talk. I'm not sure can I summarize my question, but my question is I wonder who is your main target, who is your main audience? Are scientists included them? I mean, your work is very interesting one, but at the same time, it's very detailed, very scientific. I think it's very niche, and it has a lot of context. To understand your work, people need to know science and genetical modification, blah, 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 anyway. So, but, I mean, and also your work, some of your work have critique to current science world, I think. But you're artist, so your work basically exhibited in art museum. So I wonder, do your work reach to scientists? So I'm not sure. So that's my question. So have you ever got any responses, feedbacks from scientists, except Iwasaki sensei? And is there any possibility that you respond to their feedbacks? That's my question. Thank you.
Yeah, no, thank you, Sakuragi-san, for your great question. Of course, so maybe the reason why I studied art was actually so Professor Peter Weibel, who's a very eminent media artist who was very much involved in the early Ars Electronica. So he claims he invented the word bioart in 1984. So he's a very, very important person who was always have this discussion with artists and scientists and kind of creating, and philosophy, right, and creating interesting things. And for me, using art or media art especially was just because there's the most freedom, right? So this is why I became an artist, because it allowed me the most freedom to do what I'm interested in, right? But also for me, all art is media art, right? So I never had any interest in creating aesthetic experiences or painting or anything like this. So I was always interested in how media evolved, how media emerged, and what are the possibilities of emerging media. right? So maybe this is very much late 90s. So when the internet, the computer itself became a medium, what does it mean if it became a medium, and so on and so on. And I'm very much curious how. So a lot of my research is actually about my written research is in media theory, right? And how and if biology can play a part in this media theory. But yeah, coming back to your question about who's my audience, I don't have a clear idea about it. So, but I try to do things that are interesting to me. And if other people are also excited about it, I'm very happy. But I don't target only scientists, so because we have still this how shall I say? So there's a lot of agendas of also science universities to do outreach, to promote science and art. They call it science and art, not art and science.
I know well. Yeah.
And usually science and art is just either pretty pictures which are taken in a lab, or some, let's bring an artist in the lab. He should make a picture, And and artists are not there to create pictures, and scientists are not there to do some technology stuff, So there needs to be a different conversation between that. No, but of course, I also got many feedback from other scientists, so especially in the UK. So the Imperial College was just next to the RCA, so we get a lot of feedback and a lot of, actually, I talked a lot about the previous, so the scientists there were also very, very approachable, and they were very happy to talk to artists and explain, and there are many examples. And I think if you talk to people like Joe Davis, who's one of the most interesting and amazing bio artists, he's actually scientist at the George Church Lab at Harvard, right? So he has the ability to talk to both artists and scientists on equal level, and this is kind of my goal as well, to be able to speak both languages.
And also some, and it's very interesting and a good question, which I'm also considering a lot about.
You can talk to yourself. That's okay.
No. But anyway, so of course, some scientists are also very interested in very curious people, and also are interested in art. And once I had the guest lecture by scientist who is a synthetic biologist, and from Tohoku, Tohoku University, and he started his lecture by showing George's art, and George and the Shiho Fukuhara is part of the work, and without knowing it, the Shiho attended the class. So and the scientist didn't know anything about the figure of the artist. So it's by chance, and the scientist met the Shiho and in the class, and
that's interesting.
It's a really interesting case. But so we have multiple ways for interaction, and even though it sounds very hidden to the scientists, but there's still many connections. Like actually, Anita-san attended some events, and then got Jewish like this, and it's a hope. But it's a really great question, and it's very important, and every artist should have some their own opinion, and
thank you both. And Georg-san, don't misunderstand. I'm not criticizing you.
No.
I just
please criticize me more. Please criticize.
No, sorry.
Critique.
I just hope, as now Iwasaki-sensei talked, that I hope that more space where scientists and artists and other people meet each other, interconnected,
Right, definitely.
and stimulate each other. Like, yeah, not only metaPhorest and BioClub. Yeah, that's it. Thank you.
Thank you very much.
But yeah, exactly at BioClub. So we're trying to do things. So on the one hand, educate people a little bit about biology, right? So they get a basic understanding, but also bring scientists in to understand more about art and design, right? So it goes both ways. It's not just outreach. It's just really meeting somewhere in the middle. Because I think as little understanding of what's happening in new technologies is necessary, right? So you cannot. So if you want to participate, if you want to join the discussion, everybody should know what CRISPR is and what CRISPR can do, right? So I think, but it's not rocket science, yeah. It's everybody can understand that.
Thank you. So very encouraging question. ということで、そろそろ時間かなと思うので、もし、if any other questions, and we will close the session. And thank you very much again, and George, for the great talk, and also for great questions and discussion, and for everyone. ありがとうございました。So, bye bye now, and have a nice weekend. And do you have,
and please get in touch with me on email or Facebook or Henry, we're connecting on TikTok. That's fine. But anything else is fine as well.
小木島先生いたんだ。小木島先生、なんか最後に言ってください。
very great discussion. So that, thank you very much for the great talk for Georg and Iwasaki sensei. Thank you.
Thank you very much. またもっと話しましょう。
はい。
Thank you.
じゃあ、I go and have lunch now.
Lunch time. Yeah. Okay. Thank you very much. Thanks, Oliver. Okay. Okay. Bye bye now.
Okay. Thank you. Bye bye.
Thank you.
ありがとうございました。
ありがとうございました。
ありがとうございました。Thank you.
そうですね。